Hi, my name is Dylan James, and I decided to do my PowerPoint presentation on a paper that I found on jimmunal.org entitled Human CB8 plus T cells exhibit a shared antigen threshold for different effector responses. Quite a mouthful, but pretty par for the course in any sort of peer reviewed journal for medical stuff. So basically, what is this paper about? This paper first talks about like how and what are T cells and why they're important to our body, and the role that they play in our immune response, and how important they really are when it comes to anything that dealing with any pathogen that could cause us harm. Secondly, it talks about the original consensus on how these antigen thresholds for effector responses was originally thought to have worked, and how these results either support or go against the original idea, and where do we go from here. And lastly, it talks about the results of the study and how they compare with the previous consensus from an empirical standpoint. So, what exactly are T cells? Well, T cells are the first line of defense against pathogens in the entire immune response for the human body. Essentially, they recognize different pathogens through these AGs to produce different responses on how to deal with the threat. So, if you get infected with something, that AG will tell us that will start an effector response, which will then start the process to create T cells, and the T cells will go and fight whatever the infection is, or virus, or whatever is causing you problems, and try to get rid of it and get you healthy again. And lastly, they probably serve the single most important role in our bodies have to build up immunities towards any of these pathogens that can cause us harm. They're pretty much the sole reason why we're able to have these diseases that 150 years ago would kill us if we, contracted it, to now getting just minor like fevers and coughs and maybe a bad, maybe you're in bed for a week, but now it used to kill us. So, talking about antigen thresholds and how they really function. The way T cells activate is that they recognize AGs and then are able to do with that information with what the AG tells it to deal with the threat accordingly. That they could either deal with it by usually killing or producing cytokines to deal with it in that way to get it out, and so it can no longer harm you. Until recently, the general consensus on how these T cells worked and recognized this effector was responsible by the activation AG itself. So this suggests that there was some sort of pathological memory built into the AG that would be like, so the AG would be, I've seen this before, I know what this is. Hey, T cells, you know how to deal with this, go do it like that. And what that basically means is, it would get to the point to where these T cells, these AGs would say, hey, you've seen this before, we don't need to work that hard, just, go deal with it. Or there was like, hey, this is very dangerous, we need everyone here, get in here, make all of the T cells we can. And so overall, it's just basically different pathogens will produce different responses for the T cells. Experiment that was done that these immunologists created were basically designed a systematic experiment to measure the responses of these T cells, specifically CB8 T cells, by blasting them with different stimulus that should in theory cause different results, and like the affinities that it would have. The idea was that if we measured a higher AG affinity, then that would mean that the AG was telling it, hey, we need more T cells, and if we saw lower AG affinity, it would mean we don't need as many, this isn't that dangerous. And that's what they were looking for to go into support of the previous understanding of how we thought this worked. The results of the experiment. This is where it gets kind of interesting. The results showed less of informed response of the AGs knowing exactly what it was dealing with, to a much more of what they called a digital response, where it was either all on or all off. It was either a 1 or a 0, and that we saw pretty much the same affinity over all of these iterations for this entire experiment, and this really kind of blew a hole in what what was the previous understanding, that there was some sort of memory that the AG had that would gauge the result based on the memory of the pathogen. And more importantly, and probably the most interesting part, is that this suggests that there's some other rate limiting step in these effector responses. So that is limiting the production of T cells. So we get to the conclusion of the paper, as discussed, that there was no real evidence suggesting a difference in the AG affinity among all of the 25,000 durations, showing that it was pretty much just the same response regardless of whatever they blasted it with. Secondly, it shows strong evidence that the previous consensus on how this worked was incorrect and not entirely complete. And third, it shows that there was some sort of rate limiting step that we could find, and if we could find a way to manipulate that or to stimulate that, then we could create more T cells. We don't necessarily need to mess with the effector itself. We just need to find that limiting step, and hopefully that could solve the problem for us. Anyway, overall, very, very interesting read. Kind of long, a lot of big words. But it's very interesting from an immunological standpoint, especially when it comes to the point of the type of people it could help. Anyone who is severely immunocompromised or has to be immunocompromised, or something like Crohn's disease, that are needing transfusion so their immune system is shot all the time. If they could find some way to stimulate the T cell response to get to the point to maybe help all these people, or maybe AIDS patients or HIV patients, to help their immune system, it could be really groundbreaking in this field, that if they could find some way to practically use this. Anyway, thank you for listening. That was my presentation, and have a good day.